Hello and welcome to today's session, part of Agilent Cell Analysis Global Conference. My name is Bahar Easy and I'm Seahorse Instrument Product Specialist in New England and East Canada. And with great pleasure, will be your host for today's seminar. Before I introduce our guest speaker, allow me to share a bit about Agilent and the products within Cell Analysis Division. Agilent, as a leader in life sciences, has been focused on enabling our customers to gain the insights they seek. Agilent's rapidly growing cell analysis portfolio enables deeper, more reliable insights across a variety of cell analysis applications when the investigators and drug developers seek to understand the dynamic cellular environments and the interactions. This is critically important for advancing applied translational research on vaccine development and studying autoimmune diseases, where a more comprehensive understanding of host pathogen effects and mechanisms of immune system function and response is required. Our presentation today will specifically speak to how our solutions are being employed in that context. With that, I'm pleased to welcome Dr. Heather Smallwood as our guest speaker for today. Heather is an assistant professor in the Department of Pediatrics and the Institute for the Study of Host-Pathogen Systems at the University of Tennessee Health Science Center. She is a cell biologist trained in biochemistry and infectious disease. Heather received her PhD in biochemistry and applied environmental toxicology at Washington State University, affiliated with Pacific Northwest Labs. She did her postdoctoral training in immunology and virology under Dr. Peter Doherty and Paul Thomas at St. Jude's Children's Research Hospital. Her current research focuses on defining the role of metabolism in respiratory viral infection and its regulation to identify metabiomarkers for risk assessment and therapeutic targeting. Before I hand it to Heather, let me remind you that there will be a question and answer period immediately following the talk. And to help us to get as many of your questions as possible, we encourage you to type in your questions during the presentation. We will also meet in the networking lounge at the conclusion of this webinar to further discuss topics presented today with additional experts from the field. Looking forward to our great discussions during the Q&A and in the networking session. With that, I will pass it to you, Heather. The podium is yours.
Thank you. Thank you for inviting me. And can everyone see my slides? All right. Can you see my slides? All right, I will just assume yes. So thank you for inviting me, and I'm excited to tell you a little bit about our work today. My lab, as she mentioned, is primarily focused on understanding how respiratory viruses reprogram cellular metabolism and host metabolism in order to basically be able to find universal targets. Our hope is that virus by virus, we can find targets that are changed in terms of controlling host metabolism in response to viral replication, and then eventually be able to find a universal target. And that is based on this sort of general idea that if viruses induce these metabolic reprogramming, we can actually exploit that, reduce their replication, and perhaps ameliorate disease symptoms or severity, or even prevent them altogether. So I'm going to tell you about this work in three parts. You can see on the bottom of the slide a little bit about epithelial, respiratory epithelial cell metabolism, some innate cell metabolic responses, and then some work we're doing in pediatric patients. So I probably don't need to emphasize too much here that influenza is a major problem, especially in reference to the current pandemic. All of you have probably have already had influenza. You're perfectly aware of the symptoms. You may not know how many people it affects each year. 5 to 20% of the population contracts the flu each year. And that is particularly a problem for young and the elderly. The young are generally naive to this to infection. It can hit them harder. And then, of course, flu is a particular problem impacting elderly that might be immune compromised. The vaccine is imperfect, as we all know, and our current drug therapies are that target the virus lead to resistance and have a very short treatment window. Unfortunately, influenza has a huge amount of symptom similarity with COVID-19. As you can see here from the list. One distinction to note is the change in loss of taste or smell. And I also just had to put in a word here that we are certain now that children are super spreaders of COVID-19. That was controversial for a while, but we now know that they are spreaders and that they contain more viral RNA in their upper respiratory passages. We are currently working to understand the metabolic responses of COVID-19, but I don't have enough data to be able to show you anything reliable on that today. So we're going to focus mainly on the flu and other respiratory viruses, and just know that we are working hard to determine if the same things are happening with the current virus. So if you're not familiar, following influenza infection or viral infection in general in the respiratory tract, you have a response that mediates cell migration from various compartments in the body to come in and eliminate the virus in the respiratory tract. And there's typically different kinetic curves that occur. You can see these different cell types that we have. This is from a review that we submitted, and you can just see all these different cell types have different kinetics in terms of proliferation, and the little black dots down here are representing the flu. So once we have a certain number of immune cells present, then we can knock down the virus. And some of these cells have been fairly well characterized in terms of this new field we call immunometabolism and understanding the metabolic response of different types of immune cells. Whereas some are less characterized, but at this point, T cells are fairly well understood, and their different metabolic parameters are known. And that relates to this general observations that clinicians had of observing flu or flu vaccines induce these hypermetabolic lesions, sometimes in the lymph nodes. And they were detected by PET scan, which measures an FDG or glucose analog uptake in the body. And so at that point, this was primarily attributed to these proliferating T cells, but some work I'm going to show you indicates that epithelial cells can contribute as well. And it's just a reminder, respiratory epithelial cells far outnumber immune cells, of course, in the respiratory tract. And there are many, many cells in the respiratory tract that I'm not going to cover today. This figure is taken from a review. You can see the reference down the lower right-hand corner. The first half of my talk, I'll probably go over fairly quickly because this data is published in orange here and was published in 2017. But in this first part of the talk, I'm primarily focused on these ciliated respiratory epithelial cells and respiratory epithelial cells in general, and their metabolic response to influenza infection. And we'll touch a little bit more later on some of the responses that we see with other immune cells in this area. And we're also interested in my lab in understanding how the metabolite gradient is changed following infection. And that's going to be present in this pericellular liquid that I underlined over here. I think I may mention a little bit about glucose transport in there, but we won't go into that into too great detail there. So respiratory cells far outnumber immune cells. And when we looked at immune-compromised children that had respiratory viral infections, indeed we found that they had high levels of glucose uptake in their lungs, or what is referred to as hypermetabolic regions in PET scan. So the top graphs that you see on the y-axis, you see this positive in red, the positive for glucose uptake. And then across the x-axis, you see days. And we found that there's a significant correlation between the PCR positive diagnosis with respiratory viral infection and the positive rate of hyperglycolytic lesions in the lungs. So in B here below, you see these CT scans. The top row is of the patient following a clearance of influenza infection. So that's several months later. You can see the CT scan is quite healthy, the gray there on the lungs. And below that, you see these ground glass opacities you may have been hearing about lately. This is also something that we've defined with COVID-19. And so people have been discussing this sort of consolidation and opacity in the lungs, the same thing we find here with influenza. Now, moving over, you see in the PET scan, you see these dark blotches. This is where we have an increase in FDG uptake or glucose uptake in the lungs. And then when you look at the overlay, you can really see these hot spots of metabolic activity following influenza infection. So since these kids largely were leukopenic and many immune compromised, we thought that this might not just be mediated by T cell proliferation or proliferation of immune cells in the lungs. So we went ahead and we performed proteomics on normal human bronchial epithelial cells taken from child donors and infected them for 17 hours with a strain that circulated in the 2009 pandemic, the California 04 strain. And what we found in these proteomics was that by and large, over here in yellow and A, you can see that metabolic pathways were significantly enriched and surprisingly really dominated the enrichment pathways that we had 17 hours following influenza infection. And the cord graph on the right just shows you that there's a significant amount of overlap between a lot of the proteins that we identified in these different pathways and their function within the different pathways. So we felt like the response of the respiratory epithelial cells fairly early on at 17 hours after possibly one replication cycle, maybe two, is very robust. So then we moved on and we performed some seahorse assays, and I'll talk a little bit more in detail about some of these particular assays later. But in this first slide, I'm just going to introduce that what we were looking at here is just the extracellular acidification rate as a proxy for lactate production and the glycolytic pathway. And what we found was following influenza infection here in this black bar, it was very significantly increased over uninfected controls in the white bar. Now, we were also interested in understanding if this was just a response of the epithelial cells to activation. So we used poly-IC and R848, which are both similar to simulate viral infection with an RNA virus. And then we also used LPS. We also inactivated the virus so that it could not replicate within the epithelial cells to determine if the metabolic response we were seeing was in fact impacted by viral replication. So in the BPL, LPS, and poly-IC and R848, you see that they do, in most cases, increase glycolysis with these treatments, but they are significantly diminished compared to the viral infection. And then we also used oxygen consumption rates as a proxy for mitochondrial respiration. And as you can see here, we have a significant increase in mitochondrial respiration or oxygen consumption following influenza infection or treatment with any of these other treatments. Now, in panel C, we were looking at the proton production rate, which this paper is quite old, so we typically don't publish this anymore, but it mirrors what we found in the glycolysis in panel A. Over in D, you see that following influenza infection, we have a significant increase in glucose uptake, indicating that these cells are bringing in a very significant amount of glucose from the media. And this is also significantly higher than any of the other treatments that we used. And one of the first players that we looked at, well, not the first, one of the first players that we came to look at following curating all of the proteomic data was a transcription factor known as c-Myc. And so if you're familiar in metabolism, c-Myc is sort of referred to as this master regulator of tumor metabolism and cancer metabolism. It's also implicated in some immunometabolism. And we found that following influenza infection, there was this transient increase in c-Myc. We could use a c-Myc inhibitor, and as you can see in black, this dose-dependent decrease in either the glycolysis or oxygen consumption for respiration with the addition of the c-Myc inhibitor following influenza infection. And importantly, over in H is what we refer to as glutamine and glucose addiction. So commonly in c-Myc-mediated tumors and in some cancers, they become very dependent upon increasing their glucose and glutamine consumption to fuel metabolism for cell replication. And if you deplete, it's known that if you deplete either of these substrates from the medium, these cancer cells typically die. And what we found was following influenza infection, the normal human bronchial epithelial cells were also glucose and glutamine addicted. So on the far right, you see when we restrict both glucose and glutamine, we have a significant reduction in viability only in the presence of influenza infection. And this is, again, in keeping with several other observations in c-Myc-mediated tumors. So with our proteomic data and some of this information that we had indicating these metabolic changes, we set about to do a metabolic drug targeted screen for influenza infection. In this case, because we had so many similarities between c-Myc tumors, we used a medulloblastoma drug screen that was being performed at St. Jude on tumor xenografts. And I adapted it to influenza screening and looked to see what drugs would reduce viral titer. Now, all of these data, as I mentioned before, is previously published in the first part of this talk, so I'm kind of going over it quickly. It's, as I mentioned or showed before, in Cell Reports in 2017. This particular piece is not published, and all it's showing you is that we had a significant bias in our drugs. And our drug target hits. And what I thought might be of interest to this group, if you're interested in understanding viral metabolism, is that depending upon whether we prophylactically treat with a drug or therapeutically treat with a drug, we will in fact have different drug targets that are found. Sorry, I'm trying to change the presentation. So, moving on. One of the drugs that we found was this PI3K mTOR inhibitor, BEZ235. This drug was able to restore some of the signaling events. As you can see here from the PI3K mTOR pathway, these arrows indicate the different phosphorylation events that are blocked, that are induced by the virus and then blocked by BEZ235. In panel B, you can see that c-Myc expression was significantly reduced with the use of BEZ235 in the context of viral infection. And in C, you can see that unlike other PI3K inhibitors that have been shown efficacious against influenza infection, the M gene production, the viral M gene production stayed constant irrespective of BEZ treatment, indicating that this drug was simply not just blocking an entry pathway for the virus, but because the M gene was still capable of replicating, but it has some sort of downstream effect. And when we treated with this drug, again, we saw that the glycolysis increased with influenza infection in black. And then when we used this drug to treat in the red solid bars, we saw a significant decrease in not only the glycolytic capacity of the cells, the ability of them to run ATP production on glycolysis, but also in glycolysis in general, which here is referred to as the glucose-induced response because we've added a little bit of glucose to the medium in the glycolytic stress test, which I'm going to cover in just a minute in detail. So you see here that this BEZ235 was able to restore the glycolytic capacity of these cells to non-infected levels. When we took this treatment in vivo, we started 48 hours prior to infection and followed for another 5 days for a total of 7 days with an oral gavage of BEZ235. We found that there really was no difference between the vehicle and the infected mice. However, we had a significant survival benefit that was conveyed following BEZ235 treatment here in red. So you see this very significant increase in survival whereas the Black group was at about a 15% survival rate. We bumped it up to almost 60% survival with this treatment. Then complementing that, and probably what's leading to this survival benefit, is a reduction in viral titer, as you can see in D, and an increase in the overall breathing parameters by plethysmography, and the ability, what I like to say is decreased respiratory distress in the mice following treatment with BEZ235. So what I think you can see here is that what I've tried to convince you of anyway, is that influenza induces changes in epithelial cell metabolism. This happens independent of immune cells. Immune cells aren't necessary because these experiments were performed in vitro on primary human epithelial cells in isolation. And a lot of the metabolic response that we're seeing of these epithelial cells is indicative of sort of parasitization of epithelial cells, where they are reprogrammed and lose control of their metabolism, as is evident by their inability to shift their metabolic program when glucose and glutamine are restricted. And this metabolic program is essential for viral replication. And that makes sense because the epithelial cells are the primary compartment where the virus is replicating. And of course, before a virus can bud, it requires a substantial amount of biomass, primarily proteins and lipids. And this drives a sort of metabolic phenotype that is similar to some of the some cancers, where you see this sort of increase in carbon flexibility utilization and carbon commitment into macromolecules for producing things like proteins and RNA. So now I'm going to slow down a little bit because I'd like to tell you about our work in dendritic cells. And so the difference between, there's many differences between respiratory epithelial cells and dendritic cells, but for the purpose of this talk, one of the interesting components that we're looking at is that dendritic cells, when they're infected or taking on influenza, the influenza is still present, and there's some genome replication, but the viruses are not budding. It's what we call an abortive infection. So the DCs are not actually producing virus like the epithelial cells are. And there's a substantial amount of literature indicating that DCs do have a metabolic response to TLR stimulation. So we were curious as to whether the dendritic cells would have the same sort of parasitized epithelial metabolic response that we saw, or if they would have a metabolic response similar to TLR stimulation, or a response unique to flu. And these data are currently available in preprint and BioRx and accepted to PLOS pathogens. This should be out this month. So as I mentioned before, there has been a substantial amount published on dendritic cell metabolism with LPS stimulation and TLR agonist treatments, indicating that they have this characteristic increase in glycolysis and decrease or defect in mitochondrial respiration following activation. And a lot of this work has been very eloquently done by the Pierces. Here I'm just highlighting a review by Ed Pierce. And they have extensively used the Seahorse instrument to really be able to characterize these pathways, this response in dendritic cells. And we are sticking with a model system for dendritic cells of bone marrow-derived in vitro differentiated DCs from mice, and we're infecting them with a mouse-adapted strain of influenza at the same time point as we did the epithelial cells. And when we did proteomics on these cells, on the dendritic cells, we found a very similar program, as you can see from the pie charts, to what we had previously found in the epithelial cells. That is, we had a very large increase in this significant enrichment in metabolic pathways that were changing, either upregulated in red on the top or downregulated in blue following influenza infection. When we separated this into the soluble and insoluble fraction, we wanted to understand how metabolic proteins were moving back and forth between these compartments, because not only their abundance, but also their location controls some of their activity. And we see here in this very large circle up top in red, you can see that all of these proteins involved in carbohydrate metabolism are highly increased in our upregulated in our fractions following influenza infection. So we also wanted to look with the dendritic cells at flux. So we used some traditional measures where we had different substrates that were labeled, and we fed them to the dendritic cells and watched their metabolism of these different substrates and followed the products to determine if, in fact, we could pinpoint some of the pathways involved. So we have the A here in we have an increase in glycolysis as we would expect. Significantly increases following influenza infection. Not much happening in the pentose phosphate pathway in B, with a slight decrease in the TCA cycle in C, and a slight increase in glutaminolysis in D, with not too much going on in terms of our fatty acid beta oxidation. So we wanted to look closer at this, and because dendritic cells are so responsive to the TLR agonists, of course, we wanted to go back to our Seahorse assay where we could be able to look at multiple parameters simultaneously, looking at all of the our favorite TLR agonists that simulate the flu or LPS, as well as our BPL inactivated. And this was really a great platform to allow us to be able to do live kinetic measurements. And the Seahorse can be a little bit complex and equations somewhat complicated if you're not used to it. But I kind of like to just drill it down, and my preference is to sort of try and simplify these types of things as much as possible so people don't glaze over when we're talking about metabolism. And with the Seahorse, for glycolysis, you can really basically just think about that in terms of the stoichiometry of how much glucose is coming in and how much lactate is being produced. So if you're measuring this extracellular acidification and you know the stoichiometry of this is 1-to-1, then you can really be looking and measuring this in real time, what's happening to glucose in terms of glycolysis. Likewise, you can look at your oxygen consumption rates based on oxidative phosphorylation on the far right and come to a proxy for what's happening in terms of mitochondrial respiration, particularly in a combination of using some of these very well-known inhibitors of different complexes in the electron transport chain. And so these are some of the assays that I'm going to tell you about in a moment, but I would just like to remind you that we're really just talking about math that is done on a program after we apply all these when we're looking at just extracellular acidification as a proxy for glycolysis, as well as oxygen consumption for mitochondrial respiration. And what we found was not surprising in terms of glycolysis. So in A, you can see our data where we have the control in white, and then in black we have the influenza infection as well as our BPL inactivated, poly-IC, R848, and so on. And we have this very characteristic response in what's called the glycolytic stress test, where we add glucose and then we measure glycolysis, and then we inhibit ATP synthase with oligomyosin and look to see how much the cells can operate entirely on glycolysis for ATP production, and then we'll add 2DG to sort of inhibit everything and bring it down and allow us to measure that acidification in the absence of these processes. And so you see over here under A that our glycolysis was significantly increased irrespective of the agonist or the activation level of the virus. So in both replicating and non-replicating virus, we have significant increase in glycolysis, as well as all of our TLR agonists. We see that the glycolytic capacity, for the most part, was significantly increased, and only in the case of influenza infection and LPS do we have a real increase in our non-glycolytic acidification. Below that, we went and we looked at the glucose uptake just to determine how much glucose the dendritic cells were taking in following influenza infection and these other treatments. And again, we saw an increase in glucose uptake 17 hours after influenza infection or treatment with LPS. Where it got really interesting was the response in terms of respiration. So we've used oligomycin, I mentioned before, to inhibit ATP synthase, and that allows us to be able to then calculate the ATP production. You can see in the pink box. You can use FCCP to uncouple your proton transport and then sort of ramp up the system to see what your maximal respiration is and how the system can work uninhibited. And then we use a combination of this antimycin and rotenone, and that's going to inhibit everything and bring it down so that you can calculate the non-mitochondrial oxygen consumption. And for cells like macrophages and dendritic cells, or when you're working with viruses, this is particularly important because a lot of these cells are going to have a sort of reactive oxygen burst, NADPase, oxygen and other things, INOS, other things that are going to produce radicals and oxygen species, and that's going to throw those measurements off. So you really need something like the mitochondrial stress test where you can remove that oxygen consumption that might confound your measures. So at the top here, you see that basal respiration was significantly increased following influenza infection, and this was different, significantly different than what we saw with either poly-IC, R848, or LPS. All of the TLR stimulations were significantly lower respiration following treatment with the dendritic cells and distinct from influenza. Only the inactivated virus was similar to the flu. Then when we looked at the ATP production, you can clearly see that there's this impairment with R848 and LPS. And this, as I mentioned before, was found previously and the LPS response, where it does not respond to FCCP, is a sort of characteristic response of the dendritic cells, and indicating that they are uncoupled, or the respiration is at the very least some of the different complexes are not responding, and that mitochondrial respiration is not functioning properly. And you can see that across the board on all of these measures, where R848 and LPS, these TLR agonists, respond similarly in the mitochondrial stress test. And as opposed to what we see with the flu, either the inactivated or the activated virus. And I sort of boil these down to, when you're looking at your maximal respiration, is what's your limit, how high can you go, and then your spare respiratory capacity. When I give talks, I just talked about this being how close to the limit you are operating when we come in before we start. So what's the difference between your basal and your max? And then these two measures also give us a little glimpse into how healthy, we could say, if you want to anthropomorphize the mitochondria are, how well the respiration is running. And we see that in the case of both measures, LPS is significantly different than the controls, and with the flu in the case of the proton leak as well. And if you are familiar with other measures than this type of bioenergetics, then you can kind of think of the coupling efficiency as a sort of PO surrogate for the PO ratio. So it was very interesting to us because we were anticipating that we may find the mitochondrial respiratory response of dendritic cells to flu was similar to LPS. And in fact, what we found was that poly-IC and flu were very different from R848 or LPS stimulation. Then you can use a little bit of math, understanding of buffering capacity, and use the same inhibitors, and then calculate the amount of the ATP rate using a sort of ratio and looking at your oxygen consumption and your extracellular acidification. And what you see here On the right from this ATP rate assay is the mitochondrial ATP production on the y-axis and the glycolytic ATP production on the x-axis. And it gives us a very nice just kind of global overview where you can see that what's clustering together are the mitochondrially impaired R848 and LPS treatments and the viral the virus irrespective of inactivation. So then when we come down to the bar graphs below, we're basically just using, we use the WAVE software as everyone does to calculate these measures and account for the buffering capacity. And what you see is that we have a very high rate, glycolytic ATP production rate following influenza, and particularly in comparison to our other treatments. And you can see just by the order of magnitude on the y-axis that we are heavily dependent upon the glycolytic ATP as opposed to mitochondrial ATP production overall. And that the only treatment that actually increased this was the influenza irrespective of inactivation. So there's another assay that Seahorse has developed. I think we're going to hit them all probably in this talk, but and that is the flex fuel assay. And I am going to talk about this a little bit differently than how it's often viewed. And that's largely because what I was interested in understanding was not the preference of the dendritic cells for a single substrate, but their ability to work with different substrates and to move back and forth from the substrates based on our observation that I'll talk about in a minute regarding their capacity for using glucose and glutamine. And it's essentially similar to what we've been looking at in terms of monitoring the oxygen consumption rate, as you can see here underneath this little green bean mitochondria. But we're also going to add different inhibitors to stop different substrate from coming in and then attempt to answer the question, what is the capacity for the cells to run entirely on a specific substrate or their dependency on a substrate? So I kind of equate it to the ability of a person to be able to survive entirely on hamburgers or entirely without hamburgers. I know which I would prefer, but anyway, so your capacity versus your dependency with the dendritic cells, there was not really a whole lot of dependency on any particular substrate following influenza infection. But the capacity slightly increased for their ability to use any of the substrates, whether it was fatty acid, glutamine, or pyruvate to fuel metabolism. And then that basically that calculation allows you can calculate from those two numbers the overall flexibility of the cells, which was slightly increased. But I prefer to look at the capacity and dependency, but definitely we can say that influenza infection changes the plasticity of the dendritic cells in terms of their preference for mitochondrial substrates. Now, unlike the NHBs, which you can see in H here, which I showed you before, which had this dramatic 50% loss in viability following influenza infection and restriction of glutamine and glucose, you see that the dendritic cells over on the left do not have that same phenotype. And they remain viable irrespective of restricting glucose and glutamine. And we believe that is through differences that the dendritic cells have in terms of controlling their PI3K, mTOR pathway, and some other pathways involved here, where they do not actually turn on the same metabolic pathways and responses to flu that the normal human bronchial epithelial cells do. And we're still exploring some of that area. However, they do have this increase in c-Myc. This increase is later. It occurs at 4 hours, but we do have a transient increase in c-Myc in the dendritic cells. And once again, when we use our c-Myc inhibitor in orange here, you can see that we have a loss of the glycolysis and mitochondrial respiration. Those are the basal levels measured from the glycolytic stress test and the mitochondrial stress test. So what we wanted to know then was if we inhibited these different substrates or c-Myc, did it have a functional impact on the dendritic cells? And what we found was in the top panel that inhibiting c-Myc really inhibited the migratory capacity of the infected dendritic cells. So we just looked at their ability to migrate into this region following influenza infection, which is significantly increased with infection. And then when you applied MYC inhibitor, it was brought back down to normal levels. And when you inhibited other mitochondrial substrates, it did have some effect, but not to the levels of what we saw with c-Myc. We also used a Seahorse T-cell activation assay, and that's basically just working off the understanding that T cells have this rapid increase in glycolysis following activation, and you can stimulate them and look at that response immediately, which was important for us because what we were interested in looking at was the transfer of inhibiting something in a dendritic cell. How did it transfer to T cells? If we looked at that at a long term, say 72 hours or further out, we would lose the ability to have that pathway inhibited in the dendritic cells, and it no longer would translate. And we did do time courses on that into the responses for the T cells. So we were really interested in this assay because it allowed us to be able to do something very, very rapidly assess it. And what we found was that the response of the T cells to dendritic cells that had different pathways inhibited but were infected, so they should be capable of priming the T cells, were dramatically different depending upon whether the pathways were inhibited, whether that be glutamine in green utilization, fatty acid in blue, or pyruvate in red, or when we blocked c-Myc or glycolysis in orange. We went through then and looked at the populations and how this affected the populations of T cells. And I don't have time to go over this because I want to have plenty of time for Q&A, but basically we used an OT1 system where we infected with a virus that was very specific for these OT1 T cells, and it had an ovalbumin peptide. And then we looked at what happened when we challenged them with dendritic cells that were infected with that virus in the context of different inhibitors in the dendritic cells. And we found that indeed we had a significant loss of different T cell populations when some of the inhibitors were used, as well as the VEZ235 inhibitor. And I didn't mention this previously, but after our work in mice, the VEZ235 has since been shown to significantly reduce respiratory viral infections in the elderly. And so we're really interested in understanding how that response is different between its ability to inhibit what I showed you originally in the epithelial cells versus what it seems to be doing in our dendritic cells, and how it may be dampening some aberrant immune responses there. We went in vivo and just made sure that what we had seen in our in vitro systems were recapitulated. So we removed these highly inflammatory tip DCs from the lungs of mice 9 days following influenza infection. We did the proteomics. As you can see here, it's very similar to what we found in vitro. As I mentioned before, we were asked to do it with multiple viruses. We tried 3 different influenza strains. It all recapitulated what we were finding. And then what's important here to note is that when we compared the glycolytic versus TCA cycle proteins and expression of these enzymes from the dendritic cells that were bone marrow derived in B, or tip DCs from the lung in C, we found that they really recapitulated each other well, and we have this highly glycolytic phenotype in the dendritic cells, irrespective of whether they are coming from an in vivo infection or an in vitro infection. And the TCA cycle is not ameliorated, as we've seen before. It appears to be this sort of non-canonical and low functioning pathway, but it is significantly less than what we see in the glycolysis. So it looks like in our dendritic cells, at any rate, the metabolic response that we're seeing to dendritic cells is really more of their activation. And what's interesting about it is that it's unique in comparison to what we see with LPS, and it's unique in comparison to what we see with R848. We saw a much more similar phenotype with the inactivated virus, which makes sense since they don't replicate in DCs, and it was similar to what we found with poly-IC, comparatively speaking. And so we think that what's happening in dendritic cells, as others have postulated, is their metabolic response is occurring as a part of activation. However, this activation, at least in the case of influenza, is distinct from other agonists. So I don't have a whole lot of time, so I will tell you that this is summarized. Some of this work is summarized in a review that we recently published in Immunological Reviews, where we sort of compare what we found in the epithelial cells and in the dendritic cells. And basically, what the overall takeaway is that these epithelial cells are slaves to the virus. They're glutamine and glucose addicted. They don't have a lot of control. The dendritic cells appear to have this metabolic plasticity. They can toggle back and forth between different substrates, even in the context of activation from the virus. This makes sense. They have to move through nutrient deplete regions. They have to go through nutrient deplete areas such as a tightly packed lymph node, move through the circulatory system, which may be rich, and then move into territories such as an infected lung, which will have a foci and that we have believe will be nutrient deplete, similar to a tumor, because the epithelial cells are taking up so much glucose and glutamine. We don't have time to show you the data that we're working on to prove that right now, because I wanted to get to a little bit of clinical data. And that clinical data is just a sort of a little work that we just published in a clinical, or I'm sorry, that was just accepted in a clinical report. And that's where we are looking at the similar metabolic changes in respiratory viral infections. In this case, we're looking at respiratory syncytial virus. And our general question was, these viruses are different. A virologist will tell you that these are very different viruses, but the symptoms are very similar. And we wanted to know if we would get the same sort of bioenergetic profile from clinical samples and with RSV infection as what we found with the flu. RSV, again, induces very similar symptoms. Children get it. There's no known treatment. And I believe it's by the time of 6 or 7 years old, virtually 100% penetrance in the population of children. It can be very, very dangerous for some children. Again, we saw these hypermetabolic lesions in children with RSV infection via PET scan, where we have this these ground glass opacities in the CT scan, and the PET scan showing a really high level of FDG concentration and uptake in the kids' lungs. Unique to RSV, what we found was that RSV tends to linger in terms of the hypermetabolism. We appear to have this a significant increase, if you look in the graph on the right, in terms of the PET scan positivity rate and the days. Is very different from any of the patterns that we find in the other viral infections that we studied, indicating that the RSV is inducing that hypermetabolism longer. So we did a pilot study. We had 6 children. We had we originally had 3 controls and 3 RSV infected children. And one of the controls ended up being positive when we did the PCR. So we ended up with 4 RSV infected children and compared them to 2 controls. We did nasopharyngeal rinses of these children to pull out the cells and the metabolites out of their upper airways during infection. We performed mass spectrometry again, in this case metabolomics, looking to see if there were changes in the metabolite pattern within the upper airways. And what we found was that there were some significant differences in common in lactate, for example, and other metabolic metabolites that one would expect based on our previous studies and some of our in vitro data. When we looked at cells, so these cells are from children's upper respiratory tract, and we put them on the XFB96 and did the Seahorse assay. We did both the glycolytic stress test as well as the mitochondrial stress test, and you can see quite clearly that RSV infection significantly increases glycolysis and the other parameters that we found similar to influenza. And these samples taken from children are displaying the similar patterns in terms of mitochondrial respiration, if not higher mitochondrial respiration than what we found with the flu. We also did single-cell transcriptomics. We're still working on some of that, but basically, unfortunately, these data came from cryopreserved cells. We weren't able to do everything in one day, and what we found was we weren't able to recapitulate some of the pathways that we had anticipated in terms of the metabolic response. So the take home from that is if you're taking respiratory epithelial cells out of children from the upper airways, you should process and use those samples quickly if you're intending on monitoring metabolism. We did see quite a few changes. Obviously, we were able to capture all of the immune compartment. The respiratory epithelial cells don't make it through cryopreservation in these types of samples, but we were able to look at the immune cells and we were able to get profiles of them, but we do not believe that the metabolic responses that we saw were indicative of what we would find in a fresh sample. So we're repeating those experiments. So indeed, RSV induces similar glycolytic and mitochondrial bioenergetics. Is what we found in vitro in the flu and in previously in general. So we think this may be a sort of broad response to respiratory viruses. As I said before, we're looking in SARS-CoV-2 right now. I'll just conclude with basically, we find these respiratory viral infections increasing metabolism in the lungs as well as in these upper respiratory samples. It seems like we can modulate it. It appears to be cell-specific with the modulation in terms of whether we use PEZ-235 on epithelial cells or innate cells. They have very different responses to the drug. And because there are inherent differences in pathways, these two cells have inherently different responses and abilities to control their metabolism in the face of influenza infection. We are currently exploring how neighboring cells impact that and how the metabolite milieu impacts that. I'd just like to acknowledge my lab. We are hiring, and these are the ladies that have done all of this work and some of the other people working with me on the various studies. So with that, I'd be happy to take any questions.
Thanks, Heather. Fantastic presentation. We will now start the Q&A portion of the webinar. If you have any questions you'd like to ask, please submit it. We'll be able to get to a couple of questions before the end of the hour. However, even if we don't make it to your question, we'll be forwarding all of them to our speaker and the Agilent team to follow up with you. Let's get started. And the first question that we received, Heather, is, do you think that the lack of effective viral resolution or increased risk in patients with metabolic diseases such as obesity or diabetic patient for flu or COVID could be related to an altered metabolic flexibility in dendritic cells acting as APC that reduce T cell activation?
So I think that there's a lot of work to be done. So I'm not an obesity researcher. I am dabbling currently in looking at some of that. But also, let me just say, I can't actually see the questions, and I wasn't able to see them during my talk. So I'm going to try and go back. Yeah, I still can't see question number one. But yeah, I think that certainly especially with, well, let's go with diabetics, because I think that's a simpler case. There's a lot of argument to be made for the amount of glucose impacting and perhaps hyperdriving some immune response to something like influenza in the upper airways, just based on the knowledge that we have so much higher glucose levels in that fluid that I discussed that I wouldn't be able to cover at the beginning. And an imbalance in the metabolite gradient, the transepithelial metabolite gradient, and an imbalance in high levels of glucose, I think, in the lungs in general, are going to increase your disease pathology probably through not just dendritic cell antigen presentation, but through amplifying all the way through from the innate to the adaptive response, as well as probably leading to a higher level of viral replication. So not only are the metabolites feeding a higher level of virus probably in that area, they're also feeding a higher level of immune pathology.
Perfect. I'm going to combine two questions for next one. And I'm hoping that we can get to both. Did you check the proteomics and the genomics pattern after viral infection? And have you considered using Seahorse for low throughput compound screening to orthogonally confirm your proteomics results? You've shown it for both the epithelial cells and dendritic cells.
So we have not use the Seahorse for confirming the proteomics. So that's what you just asked me?
Mm-hmm.
Yeah. I mean, beyond the pathway analysis, right? So we knew beyond a general view. So in general, from the proteomics, we saw this sort of characteristic Warburg-like metabolism, which is, of course, you can capture in the Seahorse. But in terms of specific targets, no, it was just a broad overview. Does that answer your question?
Yes, it did. But do you envision Seahorse to be useful in that direction?
Where I find it intriguingly useful for my work is that what we were able to see in terms of our drug responses, the response that seems to be that the drugs that we found. Now, I have to preface this with I only took 3 of the main drugs in vivo, but the successful Seahorse drug was also the successful drug in vivo.
Oh, okay. Good to
we
hear.
were looking at metabolic targeting drugs. The drug that restored our metabolic program in the Seahorse was also the drug that responded in vivo. Now, whether that's just, luck, I don't know yet.
Going back to the first part of the question, were you able to do any genomics or, you've shown some of your proteomics data upon infection, but was there any correlation between the genomics and proteomics data?
So are you talking about the transcriptomics in the TIP-DCs versus the proteomics?
Yes. It was one of our audience's question, but I'm assuming that's yes.
So we have looked at that, and if you're asking me if I have looked at sort of a one-by-one to see how well is the expression translating to the protein, no, I have not done a within group comparison. We use the transcriptomics on the TIP-DCs primarily because obviously in vivo we get much less, so we're not going to be able to do the proteomics that we could do with our model systems. However, you could see from the graph that I showed that in general, when we were looking at the changes of the proteins or the transcripts, they were trending in the same direction. So the transcripts for the TIP-DCs that for glycolysis were significantly higher than TCA cycle, as well as the proteins that we saw in the bone marrow-derived DCs. Glycolysis significantly higher than TCA cycle.
Fantastic. Next question is, there have been repeated reports suggesting virus-induced mitochondrial dysfunction. Is there any implication of flu-caused mitochondrial dysfunction in increased glycolytic energy production for flu-infected epithelial cells, in contrast to TLR activation by the agonist or inactivated virus?
So I think, I don't know whether they're specifically talking, there are flu, so flu technically, one of the reasons for using flu in this particular work, aside from the fact that I'm very interested in influenza, is that it's excellent because it doesn't have a lot of genetic components. Other viruses like CMV, there are other viruses that carry known metabolic regulators, known proteins that are going to be involved in them. Flu does have it does encode for a protein that localizes the mitochondria and is thought to have some activity there. In terms of the epithelial response, the data that I showed with the, sorry, I'm looking at it on the screen. The data that I showed with the epithelial cells following infection would indicate that their oxygen consumption rate, respiration, is still functional. So at least at 17 hours in this system, I don't see an evidence that it's destroyed. I would anticipate, however, at longer time points, that is what part of what's mediating epithelial death. Because of course, after so long, they can only keep up this crazy metabolic program for so long, right? They're going to die, and they can't continue to bleb off their membranes and keep producing for the virus forever. And so I would assume that at a later time point, if I were guessing, you would encounter that mitochondrial damage.
Awesome. I wanted to wrap it up at this point because we are at the top of the hour, but please join us during the networking event. You can access it via the link when you close this webinar. And also there is a direct link on the main platform for the LabRoots event. There we will continue the conversation and share more questions from the field with you. Thank you, Heather, for sharing your work with us, and I would like to conclude the meeting.